Welcome to the Deep Dive. We're here to help you get seriously well-informed, fast. We sift through dense stuff, pull out the key insights, and try to make it engaging.
Absolutely.
Today's Deep Dive is really geared towards the learner. Anyone who wants that deep understanding without getting totally bogged down, loves those aha moments, and likes things, well, clearly laid out.
And today's material is definitely rich with potential aha moments. We've been digging into numerous posts from the subreddit TSoPE, that's the science of penis enlargement.
Right.
Now, putting the name aside for a moment, the community there actually does these incredibly detailed breakdowns of erection biochemistry, and crucially, how things like supplements and medicines might optimize function.
Okay.
Our focus is really on the work of contributors like Karl and Semtex, particularly from their wiki project. Got it. So the mission today is pretty clear. Understand the biochemical roadmap for an erection. But more than that, right? We're looking for the potential levers. The sources talk about nutrition, supplements, pharmaceuticals, ways to potentially influence these pathways.
Exactly. It's all about understanding the science presented in these sources, identifying those levers.
And just to be clear for you, the listener, while the source is a community focused on, well, enhancement, we're focused squarely on the science they present, understanding the mechanisms.
That's the core goal.
Okay, let's start at the beginning then. This whole biochemical cascade for an erection, where does it physically kick off?
It all starts upstream with neural signals, brain down to the spinal cord. The sources really map out two main routes here.
Two routes.
Yeah. First is the brainstem pathway. This one's largely behind nocturnal erections, the ones during REM sleep.
Ah, the morning phenomena. Okay, biochemically speaking, what's happening there? Like, what's a switch? Well, during REM, you get this increase in cholinergic activity. I think acetylcholine in the brainstem. At the same time, adrenergic and serotonergic activity decreases.
So less stress-related signals, more rest and digest.
Exactly. Sympathetic tone goes down, parasympathetic tone ramps up. It creates the right internal environment.
Makes sense for sleep. And do the sources mention ways to nudge this pathway? Any levers here?
They do. They talk about choline precursors, things like alpha-GPC, acetylcholine. Big point is they cross the blood-brain barrier, so they can potentially boost acetylcholine production directly.
Okay.
Also mentioned are acetylcholine esterase inhibitors, like galantamine, they stop acetylcholine breakdown.
Yeah.
And then GABAergic stuff, like L-theanine or certain forms of magnesium, which might help enhance that parasympathetic state.
Interesting. So that's the sort of automatic nighttime pathway. What about erections triggered by, well, actual arousal?
Right, that's the second route, the mesolimbic pathway. This one fires up from sensory input, emotional arousal, think sexual cues. It engages the brain's reward circuitry, nucleus accumbens, VTA, and involves dopamine release.
Dopamine, the motivation molecule. Makes sense for arousal. What happens after the dopamine hits?
Those signals are processed in the hypothalamus, specifically the MPOA, the medial preoptic area. Think of it as a control hub. It integrates the cues and then triggers parasympathetic activation down in the sacral part of the spinal cord, S2 to S4.
And the levers for this pathway? More dopamine-related stuff?
Seems like it. The sources mention dopamine precursors, L-tyrosine, or mucuna pruriens, which has L-DOPA. Also, dopamine reuptake inhibitors like selegiline, though noted as off-label use.
Right.
And then dopamine agonists, things like pramipexole or cabergoline. But the sources definitely add a note of caution there about potential side effects with those stronger ones.
Okay, so we have the nighttime brainstem pathway and the arousal-driven mesolimbic pathway. They eventually meet up, right? Where's the rendezvous point?
They both converge down to the sacral spinal cord, S2 to S4. Both ultimately lead to increased parasympathetic output from there.
And from the spinal cord.
Preganglionic neurons take the signal via pelvic nerves to the pelvic plexus, sort of junction box. There they connect with postganglionic neurons.
Okay.
And those postganglionic fibers bundle together to form the cavernous nerves. These are the nerves that directly innervate the crucial parts, the smooth muscle and arteries inside the penis.
Right. Getting down to the business end now. What happens when those cavernous nerves get the signal? What do they release?
Two key players get released. The big one is nitric oxide, NO. It's made right there by an enzyme called neuronal nitric oxide synthase, or NNOS.
Nitric oxide. Okay, that name definitely rings a bell. Sounds central.
It really is. And the other important messenger released is VIP vasoactive intestinal peptide. It assists the process.
So focusing on NO, since it seems so key, according to the sources, are there ways to boost it at this stage, make more of it, or make it work better?
Absolutely. The sources list several approaches. NO precursors are a big one. L-arginine and L-citrulline.
Heard of those.
Then there are things that enhance the enzyme making NO, like BH4, or substances that inhibit things that block NO production, like ADMA inhibitors.
Okay.
Minerals like selenium and zinc are mentioned too for supporting NOS function. And interestingly, things like ACE inhibitors or ARBs, blood pressure meds, get a nod because they can indirectly help by improving the health of the blood vessel lining, where another type of NO synthase lives.
Wow, lots of potential inputs. And VIP.
Boosting VIP directly seems more speculative, kind of an area of ongoing research according to the sources.
Okay, so NO is released from the nerves. It gets to the smooth muscle cells in the penis. What exactly does it do there? What's the molecular magic?
Right, now we're inside the muscle cell. The main pathway described is NO, activating an enzyme called soluble guanylate cyclase, SGC.
SGC, got it.
Think if SGC is a switch, NO flips it on. When it's on, SGC takes GTP, an energy molecule, and converts it into cyclic GMP, or cGMP.
cGMP, another key acronym. So NO makes cGMP.
Exactly. And cGMP is the internal messenger. It carries the signal forward inside the cell.
What instructions does cGMP give the muscle cell?
cGMP activates another enzyme, protein kinase G, or PKG.
Okay.
And PKG is the real workhorse for relaxation here. It does several things. Opens channels so potassium ions leave the cell, which helps relax it. It inhibits channels that let calcium in. It ramps up the storage of calcium inside internal compartments.
So basically, it's trying to lower the active calcium level in the main part of the cell?
Precisely. Less active calcium means less contraction. PKG also directly inhibits the enzyme that causes contraction, which is myosin light chain kinase, MLCK.
Wow, okay.
Yeah.
Quite a cascade. The end goal is just lower calcium, inactive MLCK, muscle relaxes.
That's the essence of it.
And what about interventions targeting this specific NO-cGMP PKG stage? I bet this is where the famous drugs come in.
You nailed it. This is prime territory for PDE5 inhibitors. Sildenafil Viagra, tadalafil Cialis, also others mentioned like Ecarin and Vidavanafil.
Here they work again.
They block the enzyme, PDE5, whose job is to break down cGMP. So if you block the breakdown crew, cGMP sticks around longer, keeps PKG active longer, and maintains relaxation.
Ah, okay. Keeps the signal going.
Right. The sources also mention NO donors, things that directly supply NO, like beetroot, arugula, or even certain medications, and ROCK inhibitors, like Fasudil, which block a different contraction pathway. Even statins get mentioned, likely for their broader vascular health benefits, improving the overall environment.
So that's the main highway, the NO pathway. You also mentioned a helper pathway involving VIP?
Correct, the VIP to CAMMP pathway. It's seen as more of an auxiliary route. VIP binds to its own receptors, VPAC receptors on the smooth muscle.
Okay.
This activates a different enzyme, adenyl cyclase. And that enzyme converts ATP into cyclic AMP or CAMP, another second messenger like cGMP.
So CMP, does it also promote relaxation?
It does. CMP activates protein kinase A or PKA.
Similar to PKG.
Yeah, similar actions. PKA also inhibits MLCK, the contraction enzyme. It boosts calcium pumps to sequester calcium away and helps open potassium channels. All roads lead to lower intracellular calcium and relaxation.
And interventions for the CAMP pathway.
Fewer mentioned, but forskolin comes up. It directly stimulates adenyl cyclase to make more CAMP. And pentoxifylline, which is a broader type of PDE inhibitor that can help CAMP levels.
Okay, so whether it's through cGMP or CAMP, the smooth muscle gets the message. Relax. How does that translate into the physical erection?
Simple mechanics, really. When that smooth muscle relaxes, the arteries supplying blood to the penis widen dramatically. That's vasodilation.
Right.
Blood rushes into the spongy chambers, the corpora cavernosa. They fill up like balloons. And as they expand, they press on the veins that normally drain the blood away, compressing them.
Trapping the blood inside.
Exactly. Increase inflow, decrease outflow. That pressure buildup is what creates rigidity.
The grand finale, any interventions mentioned that work right at this stage? Maybe protecting the whole system?
Yeah, antioxidants are mentioned here. Things like NAC, vitamin C, vitamin E. The idea is they protect NO and cGMP from being degraded by damaging molecules called free radicals, or ROS. Keep the keep layer safer.
Okay.
And some general vasodilators, like yohimbine, are also listed, potentially helping with that final blood flow aspect.
This is fascinating. We've mapped the whole journey from a thought, or sleep, down to the final result. Okay, let's zoom back in on some of those key levers, those interventions the sources highlighted, starting with the big ones. PDE5 inhibitors, Viagra, Cialis.
Right. So as the sources really emphasize, tadalafil and sildenafil both work the same basic way. inhibiting that PDE5 enzyme.
The one that breaks down cGMP.
Exactly.
Hmm.
But the key takeaway, and the sources are clear on this, is they don't cause erections out of thin air. They enhance the natural response to sexual stimulation.
Oh, so.
By stopping cGMP breakdown, they make the relaxation signal produced by NO much stronger and last longer. It's like they turn up the volume on the body's own signal. You still need the initial stimulation, the NO release, for them to have something to amplify.
That's a critical point. You need the arousal first. They just make the response more robust.
Precisely. And speaking of arousal, the sources briefly touch on the brain's role beyond just the initial trigger, right? The complexity of it all.
Yeah.
Mentioning areas like the amygdala, hippocampus, hypothalamus, prefrontal cortex, all involved in processing cues, memory, emotion related to sex. Specific spots like the MPOA, INAH3, PVN, and the hypothalamus are key control centers. And dopamine's role in the reward centers, nucleus accumbens, driving that motivation. It's a whole brain-body connection.
It really underscores how mental state and physical response are linked. The sources also mention the balance between the two nervous systems.
Ah, yes, the parasympathetic versus sympathetic. Crucial balance. Parasympathetic drives the erection of the rest and digest system, allowing relaxation. Sympathetic is more involved in ejaculation and returning to flaccid state, the fight-or-flight system.
So PDE5 inhibitors help keep the parasympathetic side dominant during arousal.
Essentially, yes. When stimulation is present, they help ensure that parasympathetic relaxation signal wins out over potential sympathetic constriction signals.
Okay. Now, let's switch gears to supplements. L-Citrulline and L-Arginine. The sources talked about a synergy. Why is taking both supposedly better than just taking more L-Citrulline if citrulline is absorbed better anyway?
Yeah, that's a really neat piece of biochemistry explained in the sources.
Right.
So citrulline does bypass the liver's first-pass metabolism better than arginine does.
Right.
But the synergy comes from L-citrulline's other effect. It inhibits arginase.
Arginase.
Yeah, arginase 1 and 2. These are the enzymes that break down L-arginine in your body. So by taking L-citrulline, you're not just providing a source that converts to arginine later, you're also inhibiting the breakdown of the L-arginine you do take or that your body already has.
Ah, so the citrulline acts like a bodyguard for the arginine, letting more of it survive to make NO.
That's a great way to put it. You get better absorption and reduced breakdown. A double whammy.
Clever. And did the sources mention other things that inhibit arginase?
They did list quite a few potential ones, yeah. L-norvaline. Extracts from cocoa, berberine, resveratrol, cinnamon agmatine, pacetanol, chlorogenic acid like from coffee, even nitrosygine, which stabilizes arginine. Shows there are many angles to this.
Interesting. And the ratio, citrulline to arginine.
A lot of studies used 1.1, but the sources noted some research suggests maybe a 2.1 ratio, citrulline to arginine might be even better. Still evolving, perhaps.
Okay. Next up, NAC, N-acetylcysteine. What's its main contribution here, according to the sources?
NAC's role is primarily as a protector, not a direct producer of NO. It works because it's a precursor to glutathione.
Glutathione, the antioxidant.
Exactly, a major one. And the problem is these damaging molecules, reactive oxygen species, or ROS, they love to react with nitric oxide. They basically neutralize it, form peroxynitrite, and take NO out of commission.
So less NO available to relax things.
Right. By boosting glutathione levels, NAC helps mop up those ROS. Less ROS means less NO gets destroyed, so the NO you do produce is more effective.
So it's like keeping the environment clean for NO to work properly.
That's a perfect analogy. And because of that, it creates this synergy. If you're taking things to boost NO production, like citrulline arginine, or things to stop cGMP breakdown, like PD5 inhibitors, adding NAC helps ensure that the NO-cGMP you have actually survives long enough to do its job.
Makes sense. And the sources mentioned other benefits, too.
Yeah. Briefly touched on NAC's broader roles as an antioxidant, being neuroprotective, liver protective. It's a compound with wide-ranging effects.
Okay, now for ALCAR, acetyl L-carnitine. This one seemed to pop up a lot in the sources with a pretty broad range of actions.
It really did. ALCAR is presented as kind of a multi-tool for erectile function. It seems to hit vascular, neural, and metabolic pathways.
Okay, break that down. What's its main job?
Well, its classic role is shuttling fatty acids into mitochondria so they can be burned for energy. Basic cellular fuel logistics.
Right.
But beyond that, it seems to have effects in the brain mood, cognition support, potentially through neurotransmitter effects, maybe nerve growth factor support, and it has anti-inflammatory properties too.
And the sources cited actual human studies for ED.
Yes, quite compelling ones mentioned. Helping recovery after prostate surgery, where it seemed to boost sildenafil's effectiveness.
Okay.
Also in older men with low androgens, where it apparently performed better than testosterone replacement in some measures of erectile function and mood.
Wow.
And in diabetic ED, combining ALCAR with another carnitine, PLC, propionyl L-carnitine, seemed to improve the response to sildenafil.
So it definitely seems to play well with PDE5 inhibitors. What's the biochemical reason for that synergy?
The mechanisms discussed are interesting. ALCAR appears to upregulate NOS, the enzyme making NO in nerves, so more NO production upstream.
Right.
It also seems to downregulate a contraction pathway called Rho-AROC and potentially interacts with adenosine signaling, maybe boosting CAMP2. So if ALCAR is helping produce more NO and cGMP at the start, and PDE5 inhibitors are stopping cGMP breakdown at the end,
You get a much stronger, longer-lasting signal, covering both ends.
exactly, a nice upstream-downstream synergy.
And ALCARs had other relevant benefits mentioned, beyond just the direct erection pathway.
Yeah, the sources highlighted neuroprotective and tissue-preserving aspects, potential for nerve regeneration support, relevant if nerve damage is part of the ED picture,
Right.
and inhibiting fibrosis, that's scar tissue formation in the erectile tissue itself, which helps maintain healthy function. Plus, metabolic benefits like better insulin sensitivity, improved mitochondrial function, healthier energy production, less oxidative stress overall, all good for vascular health.
And the comparison with PLC, propionyl L-carnitine.
Sources suggested they might have slightly different strengths. AL2 perhaps better for the neural side, PLC potentially more focused on vascular benefits like blood flow. Combining them might offer the broadest coverage.
Fascinating compound. Okay, last deep dive, Mirabegron. This isn't typically an ED drug, is it? It's for overactive bladder.
That's right. Mirabegron is a TA3-endorgic agonist,
Oh.
approved for OAB. But the TSoPE sources picked up on some really interesting off-label findings relevant to EQ.
Like what, besides the bladder effect?
Well, some potential for fat-burning activating brown fat, maybe even muscle growth or sparing effects. But for our purposes, the focus is on erection and vascular effects.
Okay.
The key thing is it seems to cause penile smooth muscle relaxation through pathways that don't just rely on nitric oxide.
NO independent. How?
It activates those BED3 receptors, which leads to more CAMP production, similar to the VIP pathway we discussed. This inhibits contraction. It also seems to inhibit Rho kinase, another contraction pathway. And interestingly, it appears to boost the synthesis of hydrogen sulfide, H2S.
Hydrogen sulfide, another gas transmitter like NO.
Exactly. H2S also promotes smooth muscle relaxation, partly via cGMP. Mirabegron might also block alpha-1 adrenergic receptors, which normally cause constriction. So it's hitting relaxation from multiple angles.
That NO independent part sounds really significant, especially if someone's NO system isn't working optimally.
Precisely. The sources mentioned preclinical work, animal studies with diabetic ED, where NO signaling is often impaired, showing Mirabegron improved erections, and some early human data suggesting better erection scores in men taking it for OAB, who also had mild ED.
So tell me more about the hydrogen sulfide role. How important is that?
It seems quite relevant. Activating GRIA3 receptors appears to trigger H2S production in the penis. Like NO, H2S can activate guanylate cyclase to boost cGMP. Some evidence suggests H2S might even provide longer-lasting vasodilation than NO and could potentially help upregulate NO production too. It's another layer of complexity and potential benefit.
So Mirabegron unlocks multiple doors to relaxation, not just the NO one.
That seems to be the implication from the sources, yeah, making it a potentially interesting option, particularly for those who might have compromised NO pathways.
Wow. Okay, this has been incredibly detailed, a real journey through the biochemistry. Let's try and pull out some key takeaways for you, our listener.
Absolutely. I mean, the first takeaway is just the sheer complexity, right? From the brain signals through the nerves down to the intricate molecular dance of NO, cGMP, CMP, calcium. It's amazing it works at all.
Huh. True. But the sources show it's a logical pathway.
Definitely. And the second big takeaway is identifying those potential levers we discussed, ways the sources suggest we might influence this pathway.
Right. Things like boosting NO precursors with L-Citrulline and arginine.
Protecting NO with antioxidants like NAC.
Using multifaceted agents like ALCAR, maybe with PLC, for nerve, vessel, and metabolic health.
And potentially exploring NO-independent pathways with compounds like Mirabegron, based on the source material, and of course the established role of PDE5 inhibitors
Yeah.
in amplifying the natural signal.
It gives you a map and points out specific knobs and switches along the way.
And remember, this deep dive was all about unpacking the science presented in those TSoPE posts, giving you that shortcut to being really well informed about these mechanisms.
Exactly. Not medical advice, but understanding the science.
So
So the final thought for you, the listener, is this. Now that you've heard this breakdown, which of these pathways, which of these biochemical levers resonates most with you? Does learning about, say, the arginase inhibition by citrulline, or the NO-sparing effect of NAC, or the multi-pronged approach of ALCAR or Mirabegron, does any of that spark further curiosity?
does it make you want to look deeper into the original TLPE discussions?
Something to reflect on.
Definitely.
Until our next deep dive, stay curious.